All right, well, welcome everyone to RSRT's Cure 360 webinar series. My name is Jana von Hehn, and I'm the Senior Director of Research and Clinical Strategy at RSRT. And I have a very special guest to introduce today. We have Dr. David Lieberman from Boston Children's Hospital. Dr. Lieberman heads the Rett Clinic at Boston Children's Hospital and is an expert for the Department of Neurology for their precision medicine service. He is a physician scientist by training and earned his PhD in neuroscience and completed his medical degree at Stanford University. He did an internship and residency in pediatrics and pediatric neurology at Johns Hopkins University, followed by a fellowship in behavioral neurology at Boston Children's Hospital. Dr. Lieberman leads the study team of many clinical trials in Rett syndrome, including our ketamine clinical trial, and he's also the lead investigator on our Biosensor development initiative. Welcome, Dr. Lieberman.
Thank you very much, Jana, and thank you to Monica for the invitation to share the work we're doing together.
Excellent. And our Cure 360 webinar series topic of discussion today is assessing Rett symptoms with cutting-edge biosensors and invisible technology. And before I turn the reins over to Dr. Lieberman, I just want to put this presentation in context with the larger Rett Syndrome Research Trust initiatives. So, all right. So, our Biosensor initiative is really part of a broader focus at RSRT called our Outcome Measures and Biomarkers Development Initiative. And this is really a program that's designed to eliminate or reduce barriers for pharmaceutical companies so that it's easier for them to get into the clinical trials and get therapeutics to market more efficiently and faster for our Rett syndrome population. And so this is a multi-pronged approach that is looking at various different ways to improve therapeutic development for Rett syndrome. But what I want to focus on today is really just assessing biosensors. And this is a program where we are evaluating and developing biosensors for use in therapeutic development to achieve objective, real-time data for Rett patients in the home. So to understand this a little bit, let's talk about how efficacy is assessed in clinical trials in general. So efficacy can be assessed either by measuring something very discreetly, like tumor size in an oncology study, or if you were developing a cholesterol-lowering treatment, you would measure cholesterol levels in the blood. An alternative way to measure efficacy in a clinical trial is to give questionnaires. So a clinician or a parent or sometimes a patient themselves will answer questions about their symptoms on a written piece of paper or in an electronic format. And these questionnaires are subjective, meaning that depending on who is completing the questionnaire, the results can be very different. And in the 14 years that I have been running clinical trials, a lot of them in neurodevelopmental disorders, even if if the parent of the same child, like if the mother completes a questionnaire, you'll get very different results on the answers than if the father of that same child completes the questionnaire. And so this ends up with a lot of data collection in clinical trials being kind of noisy. And it can be really difficult sometimes to reproduce results, or there's not a lot of confidence that they're going to be reliable. And so the other aspect of answering questionnaires in a clinical trial is that you're usually asked to summarize information over a period of time. So either what are you observing in the moment, what has happened over the past week or month, or something like that. And you end up with a single data point that summarizes a lot of time. And this is very standard. This is how most neurodevelopmental disorders are assessed. This is how most neurology programs are assessed, and many, many therapeutics are developed and approved in this way. But what we'd like to do is actually move the needle a little bit to try to get Rett syndrome to be more like an oncology study or a cholesterol drug study, where you have something objective that's discrete, that you can measure directly in the patient themselves, that is not dependent on the interpretation of another individual. And an advantage of this system is that you can generate significantly more data than a single time point. So if you can continuously collect data from a biosensor, you may have a data set that's 24 hours long or 48 hours long or a week or even months. And this really opens up a new avenue to do clinical research and assess efficacy in clinical trials. And some of the things that we're interested in measuring, like heart rate variability or breathing or even sleep position, those things are really difficult to assess on a questionnaire, but they can be very easily detected on a biosensor. And so that's our goal for this program, is to really evaluate and develop these biosensors so we can improve the way efficacy is assessed in clinical trials for Rett syndrome. And so based on that introduction, I'm going to turn it over to Dr. Lieberman.
All right, I'm going to share my screen here. There we go. Thank you, Jana. I'll spend the next few minutes going over devices and biosensors for Rett syndrome that we're working on as a collaborative effort here. So when we refer to a biomarker is a measurable indicator reflecting a physiological or a pathological process. And the goal of biomarker development is to identify the molecular pattern of a disorder at the outset of its pathogenesis to help improve prompt and targeted treatment and improve patient outcomes. And biomarkers can be many different things. They can be molecules like enzymes, antibodies, antigens, hormones, nucleic acids, which might go an important alteration with relevance to disorder and be clinically significant. These biomarkers can be useful for diagnosis to help detect the disease. They can be predictive to help estimate response to treatment, or they can be prognostic, trying to give us information about future disease course. Biosensors are analytical devices that are particularly effective at detecting physiological or disease-related biomarkers. And biosensors should have sensitivity and specificity to accurately detect the biomarker. So what may be detected might be something in physiologic fluids, such as cerebrospinal fluid, blood, urine, or saliva. It could be something that measures vital signs like temperature, blood oxygenation, heart rate, blood pressure, respiratory rate. And it can do these things independent of a clinician involvement. It can be done objectively and can be done, as Jana said, in the home environment, which is important in our population. It's a less stressful environment, and it can also be recorded over a longer period of time, which may provide more meaningful results than the snapshot in time we use from our questionnaires. There are many patterns of breathing, for example, that you can detect with plethysmography, and I'll describe how that happens in just a bit, but you can see there's patterns of deep breathing, rapid, shallow breathing, tachypnea, hyperventilation, apneas, and all of these things can be more quantitative than what would come out of a kind of qualitative assessment of these activities on a questionnaire. One of the early devices was the LifeShirt device. This was used by Deborah Wiesmeyer and her collaborators to look at breathing dysregulation in Rett syndrome. And there were some interesting features that came out of early studies where control individuals, when they take a breath hold, which is shown here, this is with an abdominal plethysmography band, which shows changes in volume around the abdomen and chest, that with the breath hold, heart rate goes down as a diving reflex. Girls with Rett syndrome, when they breath hold, their heart rate goes up. So there are some differences also in the type of breath holds that girls and women with Rett syndrome take. They can have more frequent and longer duration breath holds, or they can have more rapid hyperventilation periods. So this is all detected, and in this device was the LifeShirt, which was able to capture these transduced signals. I'm going to talk today about some other devices. One is the BioRadio, the Hexoskin Smart Garment, the BioStamp Endpoint, and Emerald. These are all done as a collaborative effort between Boston Children's and Rett Syndrome Research Trust. So the BioRadio was a device that we've used in the IGF-1 study done at Boston Children's. It was also utilized by the Neuron group looking at serotonin for breathing dysregulation. What the BioRadio does is it captures data from this plasmography band over a wireless streaming and recording over Bluetooth. It captures this dual band respiratory inductance plasmography, which is a reliable technique for measurement of respiratory effort and volume. And it's particularly useful in evaluating breathing irregularities. Outcome metrics such as periods of hypo or hyperventilation, apneas, inspiratory and expiratory time, cycle length, and irregularity can be distracted from the data with automated software tools. The Hexaskin is a device that we've been using in the RSRT sponsored ketamine trial. The Hexaskin smart garment is kind of a newer version of the LifeShirt I showed earlier. The device uses non-invasive biometric sensors that are embedded within machine washable, breathable, lightweight fabric. It can record and track activity, sleep, heart rate, and respiratory function. So there are plasmography bands embedded for the chest and abdomen, and these electrodes can make contact with the skin and help record heart rate and other functions. So with the Hexaskin, you can get cardiac, pulmonary, sleep data. You can get activity, motion recordings. You get heart rate variability. These are all important to look at stress monitoring. And we can look at the QRS measures, which are also important for looking at QT intervals. The continuous respiratory ventilation with the chest and abdominal plasmography bands helps you determine the breathing rate, the minute ventilation, and the maximal oxygen consumption. The activity, intensity, peak acceleration, steps, and cadence are all obtained using this built-in accelerometer. And importantly, what we're using the Hexaskin for the ketamine trial is to help learn how well it can be useful for children less than 13, since it hasn't been used in this age group before. And it also hasn't been used for individuals with neurodevelopmental disorders. So it will be important for us to validate the usefulness of this. And we're going to be exploring the data sets we've already collected so far for Rett syndrome specific patterns. The MC10 is a group that makes the BioStamp endpoint. This is an FDA cleared wearable device. It employs a wireless remote monitoring platform. It's a silicon adherent patch that can help record a number of features. And you can collect data continuously and in a wireless fashion in the home setting. These are different patch positions that have been validated by the MC10 group in the past. And it also includes a program on the cell phone that helps to transmit the data to the computer and also record video. So these strips here, these lightweight, flexible strips, they're rechargeable biosensors. They can be kept on the skin for up to 24 hours of continuous data collection. They can be placed in many parts of the body and track 44 different metrics, including activity, posture, sleep, heart rate variability. It can record surface EMG on the skin. It can record monitor limb and body movements. And with this setup, you can capture electronic clinical outcome assessments with the questionnaires that are kept here. You can get some patient-reported outcomes. They'll ask, for example, how well you slept last night, how many times did you wake up during the night, and that's all loaded into the data system along with the patch information from the biostamp. And hopefully this will help not only with diagnosis and treatment, but hopefully it'll help us more immediately with some clinical trials that we're hoping to use it for. So I'm going to talk about next is a touchless system, the Emerald device. This is a device that was developed by Dr. Dina Katabi. She's a professor of electrical engineering and computer science at MIT. And her, along with members of her group in the Computer Science and Artificial Intelligence Laboratory, basically came up with this way to use a radio frequency signal detection to see how an individual moving through space makes changes to the wireless signals around them, and then it detects those changes and with artificial intelligence algorithms extracts important information. So this biosensor can be kept in the home environment, and our hope is to try to use it to get some important information for individuals with Rett syndrome. So any slight movement of the body as someone's walking through space will provide a signal for this device. And the device is sensitive enough that it can reveal vital sign changes such as pulse and breathing rate. The power of the signal used is 1,000 times lower than what a typical Wi-Fi device transmits, so it's very safe to use in the home. The signals travel through walls just as wireless signals do. There is no video recorded, there's no audio recorded, so there's no fear of privacy invasion. And as I said, the artificial intelligence algorithm developed by the Katabi lab analyzes these waveforms and can pull out information about motion in real time. It can capture information about breathing and heart rate and even sleep stages. And I'll show you some information about that shortly. And the device basically stitches together these reflected waves, and it learns to kind of piece together what an identifiable silhouette is, so it can detect an individual patient in a room where other individuals like their parents or caretakers might be helping out. To aid in this identification of the patient, we have the participant wear a small accelerometer around their ankle, and it allows the Emerald device to create a better filter. But as you see here on these top heat maps, these devices can get an idea of what different individuals look like, and by their reflective signals identify them uniquely. The device is pretty small. It's 30 by 35 by 5 centimeters. It's about the size of a laptop. It contains these directional antennas and a motherboard to process these signals. In the lab, the grad students pictured here are sitting by the sensor sitting on the wall. The Emerald device, as I said, can detect important information about breathing, sleep, heart rate, and gait speed. And for the sleep portion, Dr. Katabi worked with Dr. Matt Bianchi, who's the chief of the Division of Sleep Medicine at Mass General Hospital, and they worked out details for the detection of sleep stages. So the device can capture information about awake versus REM, light sleep versus deep sleep as the individual sleeps. And comparing what happens with a standard polysomnogram, which is what we have in sleep labs, where we put on electrodes on the scalp and various electrodes and sensors on the face, the correspondence between what came out of the polysomnogram and the Emerald device was 80% concurrent, which is a very good compatibility. So it can be helpful. Here is an individual in the lab just sitting there breathing at rest, and momentarily they're going to take a breath hold. Here they go, starting to hold their breath, and you don't see very much by looking at the individual, but the Emerald device is picking up the duration of the breath hold here very nicely and without any wires contacting the individual. Here we have an individual walking on this predefined maze with the Emerald device sitting in another room. So actually it's just picking up his motion as he travels through space and alters radio frequency waves in the room. And the marker here showing what the computer is detecting in real time is accurately tracking his motion. So in our study using the Emerald and MC10 biostamp, we are hoping to have 2 sequential cohorts with a total of approximately 20 patients enrolled. The participants will participate for about 4 weeks. There will be a screening visit, then we'll observe them in the home, and then we'll have a follow-up phone call. At the screening visit, they come into clinic for a short time to determine eligibility. And while they're there, we're recording video while we have the participant wear the BioRadio for about an hour as we collect respiratory data. We're using the BioRadio as more of the gold standard to help us compare the pattern from the MC10. And then once they're at their homes, they'll be observed for a period of 4 weeks. The Emerald device will be shipped to the home for that purpose. Afterwards, the Emerald device is shipped back and the MC10 is shipped back, and we have a follow-up phone call. In the first cohort will be 5 participants. We're gonna look for those with pronounced respiratory dysfunction, and we're gonna have either 1 or 2 Emerald devices in the home, and we're gonna have between 3 and 9 endpoint patches for 24-hour periods, 2 periods per week for the 4-week study to help assess patch placement. And caregivers will be given daily questionnaires to track their family members' anxiety, sleep, and seizure pattern. So in cohort 1, we're trying these 4 patch placements indicated as a start. Phone app I mentioned will, as they enter in some data about how well their child slept that night, they can also use that phone to record video of any breathing abnormalities that they want to show as examples for later review. The preliminary results from cohort 1 will help us determine whether we should continue the use of Emerald, and also will help us to optimize the MC10 endpoint placement for cohort 2. And in the cohort 2, we'll have about 15 patients enrolled, those with respiratory dysfunction, tremors, seizures, or other expanded features. And again, they'll be monitored for 4 weeks. The caregivers will continue their diary with the same information as they had before, and we'll have ongoing assessment of the endpoint. The inclusion criteria, so we would like to have our participants have typical Rett syndrome. They should have a MECP2, a genetic mutation that's pathogenic. Their primary language should be English because of the questionnaires used. There should be 4 people or less living in the home to help the Emerald device distinguish the participant. Visiting caregivers do not count for this assessment, and the family must have home access to Wi-Fi. Exclusion criteria are those patients that have difficulty sleeping in the room alone, because with 2 individuals, the sleep information will be hard to distinguish between patient and caregiver. The pets in the home can disrupt the radio signal, so we would like them to at least be kept out of the bedroom during this monitoring period. If the family's planning a long vacation or is going to be out of the home much, that would be a contraindication to be included. If the individual has an implantable device like a pacemaker or defibrillation device, that would be a problem. If they have allergies to hypersensitivities to adhesives, or if they have some other medical condition that could interfere with this study or confound the interpretation of the results. The assessments are going to be done in the home. The data, though, is going to be analyzed by Biosensors. That is a data analysis company that has worked with Neuron before on the Serazotin trial. They worked with the BioRadio device, and they're going to be analyzing the data we're generating now with the Hexaskin for the ketamine trial. And we're hoping to learn how to use the MC10 detect breathing during the awake state, and pilot the Emerald for feasibility. In the future, we might be able to use the biostamp endpoint for O2 saturation detection as well once that feature is added to the biostamp. As I mentioned, VivoSense data analysis has a lot of programs that are user-friendly to help determine breath holding, hyperventilation, quantify different features of gait, tremors, seizures, and sleep patterns. This is the research team at Boston Children's that's helping the study. Lindsay Swanson, the clinical research manager, Grace Bazine, the research nurse and study coordinator, Michelle DeLeo and Shelley Coughlin are clinical research associates that helped us out. I just want to conclude by saying thank you to our patients, thank you to our families. We learn from you all the time. I want to thank our partners, IRSF, RSRT, and the Rett Syndrome Angels, who used to be Rett Syndrome Association of Massachusetts. And I'm just going to give you here the phone number for the Rett research team at Boston Children's Hospital for those who'd like to volunteer for the study or learn more about it. This was a group of patients from the clinic at Boston Children's Hospital in our pre-COVID days where we could all gather together, which hopefully we'll be able to do again soon. So thank you very much for listening.
Great, thank you, Dr. Lieberman. That was a wonderful presentation. I think we have a couple of questions. Should we hop over? All right, I'll read the first one. So to use these devices in a clinical trial for a novel intervention, such as a gene therapy or an X-ray activation, are these 4-week validation studies sufficient for the FDA?
Oh, that's a good question. We're right now just trying to show reproducibility of the data just over time. When it comes to its use in a trial, it'll be up to the study sponsor to determine the frequency and duration of recordings. You know, if you might be doing a study over months or years, you may want to just do a 24-hour block, perhaps in a 1-month interval, just to kind of check on changes in that particular parameter over time. But what's nice about these is it's not very invasive, and long-term recordings are possible.
Yeah, and just to add to that, so our philosophy at RSRT is if we're planning to develop these biosensors fully, you know, all the way up to FDA standards, that's a lot of effort and commitment and financing that goes along with it. So our goal really is to eliminate and reduce barriers for therapeutic development. So if we can assess these biosensors and demonstrate that they are easy for families to use, that they generate good data that you can analyze and get results, then the ability for pharma companies to take that device and integrate it into their clinical development programs becomes much easier. And they can actually use the biosensors in early studies to validate other results that they may get. And eventually, the more studies that they include the devices in, they can validate these biosensors through their development programs. And so our plan is to use the early stage of the feasibility and the proof of concept that this idea works in Rett syndrome, and then hand it off to pharma to continue the development. And that's why we have so many different devices that we're looking at. We have the Hexaskin Smart Garment, the MC10 endpoint, and the Emerald. And our plan is to have a number of these tools available depending on what you want to measure, the way you want to utilize the device, and that's our plan. Okay. And then our next question Is there an opportunity to use the crowd of our Rett and duplication communities to gather data really fast to hand over to the FDA or pharma and biotech so they can hit the ground running? And the answer to that right now is no, but we hope in the very near future that our plan would be to continue to assess and validate these biosensors through our registry, where people who are signed up to participate in the registry and are interested in doing studies like this, that we already have that research population ready to go. All we need to do is get some biosensors into their hands. So the ability for us to generate large data sets and potentially assess multiple different devices is going to become easier and easier, especially once we get the registry open. All right, and then the other questions here are just about getting into the study. So as Dr. Lieberman has on the screen here, you can contact the study team there. And then our final question was whether or not these devices can be used in Europe.
Well, I mean, at least I know the BioRadio device was used during the Neuron trial, and I think at least Italy was a site there. So I'm sure they have European versions for these devices.
Great. Well, thank you. I don't see any additional questions, so I think we can go ahead and close the webinar. Just want to, again, thank Dr. David Lieberman for his time today and the wonderful team at Boston Children's Hospital. It's always such a pleasure to work with you on all of these great studies and programs. And thanks so much for taking time out of your busy schedule to update us on this collaboration.
Thank you, Jana. I appreciate the opportunity.
All right.